In this video, I'll be continuing on with the cell structure topic, and I'll be going through cell transport. So, there are two categories transport can fall under, either passive or active. Passive transport is where ATP is not needed and the movement is along a gradient, whereas active requires ATP and the movement is against the gradient. I will go through the passive transports first. So the definition of diffusion is the net movement of molecules from a region of high concentration to a region of low concentration. As you can see here, it moves from high to low. Next, we have osmosis. So the definition is the net movement of water from an area of high water potential to an area of low water potential across a partially permeable membrane. Now, the difference between osmosis and diffusion is that it's the same, but osmosis is specifically for water. Hence, for water, instead of using the term high water concentration, the scientifically correct terminology is high water potential. Now, in your exam, if you are asked for the definition of osmosis, always remember to add the last bit, across the partially permeable membrane. So, pure water has a water potential of 0. To know whether the water potential is high or low, remember the more negative the number, the lower the water potential. Now, you also need to know about the effect of osmosis on plant and animal cells. In plant cells, the main thing is that plant cells have a cell wall that protects the cell. When the plant cell is placed in a container with a high water potential, water enters the vacuole of the cell via osmosis. The pressure inside the cell pushes against the cell wall, making it turgid. However, the cell wall will prevent the plant cell from bursting as the pressure stops too much water entering. Whereas if the plant cell is placed in a lower water potential solution, water will leave the vacuole via osmosis. The cell surface membrane will start to come away from the cell membrane, and the plant is described as becoming plasmalized. Whereas animal cells do not have a cell wall, so things are a bit different. When an animal cell is put into a low water potential solution, water leaves the cell, causing it to shrink. If an animal cell is put into a high water potential solution, the water enters the cell and the pressure increases inside. As there is no cell wall to protect the cell, the pressure inside rises and it can burst, also known as lysis. Now, facilitated diffusion is still passive, however certain substances cannot cross the membrane via simple diffusion, such as large polar molecules and ions, so they need some help. They use proteins to help them carry them through the membrane. Channel proteins allow ions to go through, as their charge prevents them from diffusing across. Then carrier proteins allow large or polar molecules, as well as ions, through, as a size or charge stops them. With the carrier protein, there's a binding site on one side of the membrane, and once bound, a conformational change occurs, and the protein then opens up on the other side. So the other thing you need to know about in your exam are the factors that affect the rate of diffusion. This can come up in many different questions, whether it's describing how a cell is adapted for diffusion or an experimental question on rate of diffusion. So for these application questions, there are 4 things to remember. First is the surface area. The larger the surface area, the higher the rate of diffusion. Next is concentration gradient. The greater the difference in concentration, the higher the rate of diffusion. Next is diffusion distance. The shorter the distance the molecules need to travel, the higher the rate of diffusion. And finally, the number of proteins for facilitated diffusion. The more proteins, the greater the rate of diffusion. Now, the active forms of transport. The definition of active transport is the movement of molecules across a cell surface membrane against their concentration gradient, i.e., from a region of low concentration to a high concentration using ATP. So, this is an energy requiring process and uses carrier proteins to move these molecules against their concentration gradient. The other active form of transport is co-transport. This is the coupled movement of a pair of molecules across a membrane via a carrier protein. Like in this diagram, for example, the sodium ions are actively pumped out, so using energy, creating a high concentration of sodium outside and a low concentration inside. This then means those sodium ions can come back down via its concentration gradient here. But as sodium comes along its concentration gradient, it will bring glucose with it via facilitated diffusion. But this is still an active process as the sodium ions had to be pumped out in the first place. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.